I'm very glad to introduce the next speaker, actually from the US, Dr. Gunnar Gottschalk from Simmaron Research and University of Wisconsin, Milwaukee, as you see here. And you will describe your rapamycin study in this case, right? Yes, so please, the floor is yours.
All right. Just like to start by thanking the conference organizers, again, the patients. I know how hard it is to been in this disease for a while, but I just really appreciate people coming to these and being a part of all the research studies. Again, without you all, we can't do anything. So thank you. My name is Gunnar. I'm not Swedish. It seems like it. I'm a professor in the Department of Chemistry and Biochemistry at the University of Wisconsin, Milwaukee. And I run a small not-profit in Incline, but was originally based in Incline Village, Nevada, called Simmaron Research. We're a 501c3. I have no disclosures for today. So I try to start with this first. I always forget to thank people at the end, but science is a team sport. You need a lot of people involved in this, so I'd especially like to thank my partner and CSO, Chief Scientific Officer, Dr. Avik Roy. I'd like to thank all of the clinicians who are listed here, as well as members of the lab team. We have kind of a unique intersection, Simmaron and the university. We are a part of the university, but we're kind of not. We pay to be there. It's a good setup for us because we have access to these core facilities, and we can do things that previously I think would have been very, very expensive, but with this collaboration, it gives us a lot of options. And that includes a full-on like Shimadzu omics core, drug synthesis development. Both myself and Avik were kind of more drug development scientists. That's why we've been interested in trying newer compounds, and that's what I'm going to be talking about today. Even though rapamycin isn't a new compound, it is something that we think might be able to be repurposed for ME/CFS and potentially long COVID. So there was supposed to be a big umbrella here on this first slide, and really that umbrella should represent the heterogeneity of the patient population. Everyone's going to be a little bit different. We just heard about a 30% response rate with an antiviral Paxlovid in long COVID patients. That is just the way this is. So just know going through this that as new treatments are tried, as studies are going on, it may not help everyone. But even if it helps someone a little bit, that's important. So we're going to go through really just a quick overview of ME/CFS. The mechanism, again, this is a mechanistic biomarker-directed trial. So really an exploratory pilot more than anything else. It's not confirmatory, but we'll go through the mechanism. The design and execution of this initial trial pilot, and then our preliminary findings and implications, future directions. So this is a doozy. Really understanding the defining symptoms of this illness. There's been a lot of work done over the past 50 years identifying these different criteria for patients. We know that the prevalence is quite high in the US, 3.35 to 4.3 million. I heard 300,000 in Sweden. It's really bad. A significant reduction in quality of life. These people are not tired, okay? They are sick. There's significant challenges in the diagnosis due to the heterogeneity, kind of the umbrella term. And that's, again, why looking at specific pathways, mechanisms, which, again, may not be involved in every single person, but could very well be useful in a subset of patients, is significantly important. There are currently limited treatment options, so we need to keep doing better at finding new and or repurposing, in this case, drugs. There's a lot of theories on the causes and biomarkers. People always say, well, there's no biomarkers for chronic fatigue. No, there's a lot. There's been a lot of work done. Some of our colleagues here who are speaking later today, again, they've gone through this, but first and foremost, one of them is immune dysregulation. For example, T-cell exhaustion, macrophage inflammation could contribute to the onset and progression of the disease. Viral infection and reactivation. SARS-CoV-2, that's pretty convenient because it just happened in 2020, but looking back, seeing how the beta herpes viruses and others have really kind of pushed the persistence of ME specifically prior to there being a SARS-CoV-2 is an important factor. And again, these viruses can cause, trigger this illness. We know there's metabolic and mitochondrial dysfunction. That includes impaired glucose metabolism, defective mitochondria, and that can reduce cellular energy from the mitochondria themselves. There's neurological abnormalities. Again, here in my slide, I'm showing here sort of the central world of microglia, sort of the resident immune cells in the brain, and how peripheral markers, peripheral viruses, even if they're not inside the actual blood tissue themselves, they could be hiding in some other reservoir. So it's important to think about that when you're looking at these, looking at this illness. You know, there's challenges, like I said. Some things you can find in blood pretty easily. Other things you can't find in blood so easily. You know, if you're looking for biomarkers and you want it to work, I think you do have to look in the blood because it's really the most accessible space. But again, there's limited options, and you know, hopefully new technology, new techniques can help push that. Okay, so the mechanism. How did we get to this weird drug, rapamycin? Well, you know, there's a lot of different work that we had done. First and foremost, rapamycin is actually a bacteria that was identified on the genetic bottleneck island of Easter Island. It was identified by a Canadian scientist, Professor Nath Sagal, and it was actually found in frozen soil samples that then sat in someone's freezer for many, many years. There's a great PubMed article about the history of rapamycin. But they started looking at it because, you know, there has this sort of interesting structure. And it was initially discovered as an antifungal agent, you know, from the soil-grown bacteria. And it's named after the Rapa Nui people on Easter Island. So that's where you get rapamycin from. It has two activation sites, one being this macrocytic lactone core, which is up here, there, and then this cyclic polyketide chain. That is where the action that we're going to be looking at is addressed. So the previous medical uses, this is an FDA-approved drug. It's been approved since 1999 for organ transplant rejection. A lot of times for people that are getting, you know, kidney transplants, you need to suppress the immune system. So to be clear, this is an antibacterial medicine that is an immune suppressant agent at its full dose. It's used currently for cardiac restenosis, basically coating the valves of hearts to help reduce the rejection potential. It's got a series of other indications for LAM, a lung disease. It has a lot of cancer research applications. Kind of most interesting recently has been this focus on anti-aging that many people have postulated, and that's where we got the low dose idea from. So again, in this study that I'll be talking about, we're using a dose that's about 1/10 of what is normally provided, and we're doing it once a week rather than daily. So how does it work? Well, in the context of autoimmune or of organ rejection here, you can see this FKB1 region, the part top here. It can directly interact with cyclin E, CDK212, and prevent cell proliferation. This can help with immunosuppression. It can help prevent cancer. It can also help prevent aging. But in the context of what we've been looking at, we are looking at where the mTORC1, that polyketoid region, interacts with the mammalian target of rapamycin. We have published, and I'll get into this shortly, that mTORC1 is the primary kinase responsible for the phosphorylation of autophagy-related gene 13. Autophagy-related gene 13 is critical for a process called autophagy, which is, you know, simply put, one of the best cell waste disposal systems that we have. And we found that in a group of MECF patients, we were identifying this phosphorylated version of ATG13 in the blood. So it's an intracellular process of removing toxins and other pieces, defective mitochondria, et cetera. So if you're finding it in the blood, it means it's not doing its job inside. And essentially, over the past couple of years, really starting back in 2021, we initially found this phosphorylated version of ATG13, looking at a group of validated MECFS patients compared to healthy controls. And this led us to hypothesize and understand the process of how we can modulate mTOR activity to prevent this phosphorylation that would then allow autophagy to occur, particularly in the neuromuscular junction. You know, when you have an active mTOR, that's too high, you get this phosphorylated flavor of ATG13, and it inactivates it. And ATG13 is one of the very early upstream factors associated with the autophagy coupled lysosomal protein degradation pathway. So if you're stopping it at the level of ATG13, you're going to have autophagy impairment. We've shown in a couple of different mouse models that this autophagy impairment can directly stimulate M1, sort of pro-inflammatory macrophage activations into the neuromuscular junction here where the ANS nerves are innervating the skeletal muscles, resulting in mononucleosis, inflammation, demyelination, and apoptosis, likely fatigue. When you do animal model studies, you have to think about ways to replicate specific symptoms. In our case, we were looking at post-exertional malaise. That's something that we feel we can kind of reach out and touch because it's very unique to ME/CFS and long COVID compared to other diseases. So when we built these mice, we actually had them running on treadmills, similar to like a 2-day CPEP test. And that work is all published here. We've got a series of other papers on that if anyone's interested. And this work was funded by NIH. So in the first animal model, we simply fed black mice a whole bunch of an mTOR activator, right, to replicate what we see in the patients. And that resulted in this autophagy impairment. And then in order to investigate the direct role of ATG13 in activation, we created a transgenic knockdown model of ATG13. So in both cases, autophagy is impaired. In the first animal model, it's impaired directly through chronic mTOR activation. And then in the second model here, we're seeing that by repressing the ATG13 gene, we're identifying that these animals develop fatigue. So we had been doing all this work, and this was at a conference in 2022, and some of our colleagues that we work with, we had published sort of these two papers, kind of developed this working hypothesis. And David Kaufman, who's based out of Seattle, he had told me, "Oh, Gunnar, yeah, I've been giving rapamycin to my patients with ME/CFS." And I said, "Well, I think we have a mechanism." And from that, we went from doing basic science research, which is kind of my background, to all of a sudden doing a clinical trial, at least an exploratory pilot. And this has led to what I'm going to talk about today, the process of keeping people on once-weekly low-dose rapamycin for up to 90 days, and then looking at the various outcome measure changes, in addition to identifying changes in these biomarkers, specifically this phospho-ATG13. Again, we had identified this elevated phospho-ATG13 as a potential marker in blood. So we hypothesized that since that was driven by mTOR, if we utilize rapamycin to reduce mTOR activation, autophagy impairment would be no longer. One of the good things about doing the type of study that I'm talking about here is that it's pretty flexible in its design. We did a lot of biobanking, and it's sort of feeding the next sort of generation of studies based on this patient population, where we're even seeing longitudinal changes in things like macrophage activation states, which again gets a lot of the other mechanisms that we've seen and others have talked about previously. Okay, so the design. So again, just quickly, we have observed some ME/CFS patients. About 30 to 40% of them have this sort of chronically active mTOR. This leads to the ATG13 inactivation. It results because of the kinase activity, right? mTOR phosphorylates ATG13 and inactivates it, leading to autophagy impairment. And the result of autophagy impairment is known to be mitochondrial dysfunction, cell death, inflammation. Again, we were trying to look at specific symptoms, PEM, fatigue, brain fog. Of all the symptoms that kind of define long COVID and ME/CFS, PEM appears to be sort of the most consistent. Again, understanding that we are seeing this interaction at the level of the nerves and the muscle gave us some confidence to say, okay, well, maybe this can affect PEM. And again, our animal models with the 2-day running test demonstrated post-exertional malaise after both chronically active mTOR activation leading to autophagy impairment. So the idea was to utilize, and it was kind of a WAG, a wild ass guess, that's what we call it, 6 milligrams per week of rapamycin, oral, right? and that would help reduce this chronically active mTOR, that would prevent this phosphorylation and activation of ATG13, and also reduce the autophagy impairment, resulting, we hoped, in reduced symptoms. So again, the objectives: assess rapamycin's ability to improve symptoms and quality of life. Is it safe? Is it, are patients going to tolerate it, right? And our scientific hypothesis was the direct reduction of this phospho-ATG13. So that is what our biomarker is, and that's what we became interested in. We have another marker in addition to phospho-ATG13, one called Beclin-1. Beclin-1 is known as it's an accepted kind of pan-wide marker of autophagy function. So increases in Beclin-1 are indicative of autophagy function increasing. Conversely, elevated levels of phospho-ATG13, those are associated with less autophagy function. So those come from the blood, and we can measure those reliably. So, we started enrolling patients. This was a completely decentralized study. It's one of the benefits in the States, at least. You can run a lot of these, again, outside of the clinic. This is an oral pill. It's extremely cheap. It's off patent. It's available. But we had to, we utilized the clinical experts that we know because we're trying, again, to hammer down through that heterogeneity. We did a ramp dosing period of 1 milligram per week for up to 6 weeks. In this first 36-day period, this was just to see if we needed to, if there were going to be a series of chemical sensitivities. That's a known factor with this patient population. Each individual served as their own control. This is not placebo-controlled, so take it for what it is. But the entire goal of this was to assess really the functional biomarkers in the serum and see if we could identify those changes and sort of be the cannon fodder, hopefully to be able to develop a biological marker that could help better predict and include patients in future studies. Again, decentralized, no in-person visits, and there was a lot of mobile, follow-up with mobile phlebotomy. We collected all these samples. In a post-COVID world, a lot of the companies got used to shipping like blood samples and drugs and other things, so it's been nice to have that. FedEx works pretty well. So what we were looking for measuring effectiveness and safety, we have a series of questionnaires utilizing this REDCap server. We built it in collaboration with Dr. Maureen Hansen at Cornell. These are our primary outcome measures: 24-hour survey, the Bell Activity Scale, the multidimensional fatigue inventory, the Pittsburgh Sleep Quality Index, SF36. For assessing safety and tolerability, we did a complete blood count, comp med panel. We looked at hemoglobin A1C along with lipids. One interesting factoid about mTOR suppression therapy is that you can, by chronically suppressing it, you can increase lipid levels, specifically bad cholesterol, LDL, and triglycerides. Fortunately, in our study, like I'll go through, we did not see that at this dose, but there are animal studies that have shown that. So, it's important when you're working with this drug to continuously look at lipid panels. In this current phase, it's not listed here. We also have a pregnancy test for females. They just need to take monthly. And some other sort of general inflammatory markers like HRS-CRP. Got through that. Safety monitoring techniques. Okay. All right, so here are some of the first results. This was just recently published in the Journal of Translational Medicine, so please look it up. But we had about 108 people that were consented. There was a big drop between folks that consented to be in the study and those who didn't start the drug for various reasons. One of the issues with decentralized trials, right, is that you lose some control with not having people coming in. But again, for something like this that's relatively safe, it's been used in anti-aging protocols for years and years. At the end of the day, some people didn't start because they were starting a different intervention or it just wasn't going to fit with their life. We had 86 people start. We had 16 people stop through the ramp period, 70 people that got through the T1, 36 days. An additional 18 stopped between T1 and T2, and then we had 9 stops between T2 and T3. So in total, about 40 completed. that's kind of how this goes when you're doing these trials. You know, you see a lot of people dropping out for various reasons. One of the primary issues that we had, because we bootstrapped this in trial, it wasn't funded. We just kind of made it up as we went. You know, patients were initially paying for the drug themselves. The average cost for the labs and the drug per month was about $7.50, but for some people, that's like out of reach. So like you can't run a study that way. Here's our ME/CFS patient sort of age and demographics. About 47 years old. You know, we've got approximately 68% female to male, and duration of illness was quite long. Okay, now, of our 40 people that completed this, we assessed the analyses initially and identified a significant improvement in these fatigue scales that are validated. And, you know, this was cool to see, but at the same time, understanding this isn't a placebo-controlled trial, really, you know, you really have to take this with a grain of salt. And again, our primary idea was to try to assess these levels of serum markers of phospho-ATG13 and Becklin. But it was interesting that, number one, the drug was very well tolerated in the patients that took it. You know, we did see a significant improvement, it's particularly in something like post-exertional malaise, which again was our target. Obviously fatigue, disturbed sleep. We did have some reported side effects. There appears to be some transient GI symptoms that people have really at the end of the ramp period as they're ramping up the dose. So, you know, kind of consulting with patients, say, hey, expect that you might have some diarrhea for a couple of days. Please don't quit because of that. You know, that's again, this type of study that we're doing, you know, it gave us a lot of flexibility with our ability to kind of talk patients through that. And that was really key to moving forward. Again, these folks were followed up carefully by their clinicians. So there were antibodies available for phospho-ATG13, but they weren't specific to the serine residue that we were interested in looking at. So mTOR as a kinase is phosphorylating phospho-ATG13 at serine 248. So we had to contact Thermo Fisher. They built an antibody for us that was very, very sensitive. And what's very cool, which you'll see in the next slide, is these are markers of Becklin. So this is our pan-wide marker of autophagy function. We see a significant increase in Becklin serum expression over this between baseline to T3 in these 40 individuals that completed. Conversely, we see a significant reduction in phospho-ATG13 between baseline and T3. So we've done some correlation analyses. Again, these are published. We found very nice correlations, you know, in critical areas, mapping the questionnaire data to the biomarker data. And this is really what we wanted to see. This part was completely blinded. You know, what's great about working with someone like Maureen Hansen is she's running the entire REDCap side. We have an individual running all of the lab work at UWM, and then we have sort of a data safety monitoring board that then, you know, unblinds the data. But this was very cool to see that, again, like we saw in the mice, like we saw initially, okay, you know, it appears that we have a functional measurement of autophagy improvement simply utilizing once weekly low-dose rapamycin. This is not the end of this trial whatsoever. I'll get into future directions. You know, but it was very cool to see this correlation. Okay, some of the things that we're working on now in the next phase of this is bioavailability. It was alluded to in the Paxlovid study just spoke about. You know, these drugs, and particularly the generic versions of sirolimus rapamycin, have about a 40 to 60% difference in bioavailability, which means when you take it, it's, you know, it could not be getting to the system where you need it to go to. We needed to identify a sustainable single source of the drug, which we have done. And what we're doing now is we're taking a look at actual sirolimus as rapamycin levels in the plasma. And that's because, again, there are drug metabolizing enzymes, specifically CYP3A4 and cytochrome P450, that are responsible for the metabolism of this drug. So, and if you go and do a normal LabCorp level here, or Quest Diagnostics level, they'll say, okay, well, it's always going to flag low. Well, that's because the dose is indicated for like an immunosuppressive dose, like I said, about 10 times higher. So we think in our hands, based on some of these peak trough data that we've done, we're having patients take the drug 4 hours after getting the blood draw, 4 hours after they take the drug, and then again on Wednesday. That will give us that 66-hour half-life, for the drug, and we can identify and see that there's a pretty significant drop in serum concentration. And this is pretty heterogeneous. The data isn't complete, but there's obviously some differences, right, between these people and these people. So I think moving forward, and again, one of the reasons we continued this study design is that the dose was never optimized. We don't know. If you're a fast metabolizer, you might need to take this twice a week, not once a week. If you're a slow metabolizer, you might need to take half the dose once a week. And until we kind of really understand that, I think it's going to be really hard to run a double-blinded randomized placebo-controlled trial, because you're going to have so many different people responding at different levels. So this is our attempt in this current phase and the previous phase to kind of understand where the dose should be. We had some great responders, and if you read the paper, because I'm not going to get into this, but we had some incredible responders, individuals. We had some groups of patients that responded quite well. Again, the primary symptoms that people reported feeling better with were were post-exertional malaise, not just the number of events themselves, but the severity of the events when they did occur. We think this is a good insight into the potential use of this, and it requires and sort of demands a double-blinded randomized placebo-controlled trial. And I've seen that groups like PolyBio, they're going to be starting one, or they have started one, which is good. We know that this is limited by a lot, simply because we don't have a placebo group. We have a small sample size. There are placebo effects. There are going to be symptom reporting variabilities between individuals, right? If someone is feeling initially really good, and then for whatever reason they start feeling worse, or the opposite, that is something that we need to really understand, and that can be potentially related to the complexity of the disease itself. So the implications and future directions. This is a novel therapeutic avenue. It's a biomarker-directed trial. We think with some confidence and with some further development, we can utilize and validate these findings in larger cohorts of phospho-ATG13 being elevated at baseline and then reducing over time, and conversely Becklin and other autophagy-related markers increasing over time. We did see improved patient outcomes. We had patients that were bedridden that started walking around. I will say that our kind of average person had a sort of middle-of-the-road MECFS, but again, if you can get someone 20% better, that maybe allow them to go back to work part-time or something. And that's really what we're trying to understand with these outcome measures. We're expanding the treatment options. It's definitely, there's other ways to modulate mTOR, one being metformin. That's an AMP kinase activator. That is a similar mechanism, and I think there's some more evidence from not only ourselves but others that a subset of MECFS patients has this chronic mTOR activation status. So again, if you can select for those people, right, then you can potentially weight the clinical trial, which is what we need to do. Next steps. We need to do a larger trial. We need to do a long-term effect of the analysis. So in this current phase, we're not just following people for 90 days, we're following them up to a year and a half. And this will take time, but I think it's really important, because simply if you see an improvement, what if this is only a 90-day improvement, right? Well, that's not very useful. Or what if it extends out like a year and a half further? What are we going to do with that? The other thing is that maybe you need to pulse this dose. Maybe it's like you're on for 3 months, you're off for 2 months. You're on for 3 months, off for 2 months. We did see that the most significant changes occurred at T3 after 90 days. So that's really important to know, that in a lot of other drug trials, and MECFS in particular have this phenomena of delayed response to therapy. I know for the Ampligen studies, it could take up to sometimes a year of someone being on Ampligen before they started feeling better. So we need to understand that. And then we need to further investigate this mechanism. And specifically, does it improve mitochondrial metabolism? Does it activate T cells? Does it help fix that? So just as a teaser, you know, again, part of what we've been able to do with this work and developing this biobank is correlate, right, our ELISA findings of phospho-ATG13 and these changes over time to things like seahorse analysis, which is looking at mitochondrial function. And it's been very interesting to see, for example, in this patient, you see a significant difference between baseline and T2 with respect to their mitochondrial OCR activity. So one of the benefits, again, of being at a place like UWM and next door at the Medical College of Wisconsin is we can take these samples, you know, have them shipped to the lab within 14 hours, processed live cells, and then execute something like seahorse directly. We're also taking all the plasma, all the serum, and we're doing a multi-omics approach to help correlate our findings with these initial ELISAs. But again, I think as far as identifying patients in subsets, you know, we feel pretty strongly that this serine phospho-258 is pretty important. That's kind of it. I know that was kind of fast. I really encourage everyone to read the paper. It's a lot of work to do these things, but again, I think we are making big moves for a small group that really just kind of started a few years ago. And our goal is treatment trials. We do drug development assays, we do new drug development, but understanding that developing a new drug in ME/CFS is economically impossible. Okay, there's no clear FDA approval pathway, so you can't get investment, and that's where these opportunities for drug repurposing studies, like OMF's LIFT trial with low-dose naltrexone, like that's hugely important, right? Because there's all of these things that are out there that people can try, and if you go on Twitter and you read what people are trying, and you listen to the patients, and you try to design things by listening to the patients in controlled ways, I think you can get pretty far. That's what I have. I'm available for any questions. I'll be here all day so we can talk. Thank you so much. Thank you to the conference organizers again. Appreciate it.